Usually when we talk about a medical diagnosis, there's this expectation of clinical precision.
Oh, absolutely. It feels very mechanical, right?
Yeah, exactly. It's like you break your arm, the x-ray shows that jagged white line on a black background, and the doctor just points and says, yep, there's the problem.
It's a very comforting illusion, honestly.
Mm.
We crave things that are visible. We want them easily categorized. I mean, a broken bone is clean. It's either broken or it's not.
Right. But then you step away from the x-ray machine and peer into, well, the microscopic world of infectious diseases and immunology.
And suddenly that neat binary comfort just vanishes completely.
It totally vanishes. We're looking at a diagnostic landscape today that is, honestly, a massive invisible war zone.
The ultimate muddy waters of medicine, the pathogens we're dealing with, these microscopic invaders, they're constantly changing the rules of engagement.
Yeah.
They use disguises, they hijack our own cellular machinery, and they just sort of wait in the shadows for the perfect moment to strike.
And that unseen battlefield is exactly why we are here today. So, welcome to this deep dive.
Glad to be diving into this one.
We are jumping into a mountain of fascinating source material today. Everything from how pathogens are categorized to the actual mechanics of human immunity.
There is a lot to cover.
There really is. But our mission for you isn't to get bogged down memorizing a medical textbook. We want to shortcut your way to understanding how these invisible wars are actually fought and won.
Right, getting to the actual strategy.
Exactly. We're going to decode how bacteria, viruses, and fungi operate, how they execute their attacks, and, crucially, how your immune system's very specific blind spots dictate exactly which illnesses you get.
Because getting sick isn't just a random roll of the dice, you know.
It feels like it is sometimes.
It does, but there is a deeply logical, almost highly choreographed biological interaction happening between the invader and your defenses.
By the end of this, I promise you're going to have some serious aha moments about why certain illnesses act the way they do in your body.
Oh, for sure.
So before we get into the wild, sneaky ways that viruses and fungi attack us, we really have to start with the classic, old-school invaders, bacteria.
The heavy hitters.
Right. And to even begin fighting them, scientists have to sort these microscopic fortresses into, like, biological buckets?
They need a classification system.
Yeah. Okay, let's unpack this. The very first sorting mechanism seems to be a dye test called the Gram stain.
A true classic in microbiology.
It splits bacteria into gram-positive, which stain purple, and gram-negative, which stain pink. It feels a little bit like, I don't know, sorting laundry into darks and lights.
You know, that laundry analogy is actually a great way to visualize the color split. But the clothing, in this case, is a set of biological armor.
Oh.
The Gram stain isn't just a fun cosmetic preference for the bacteria. It reveals a fundamental structural reality.
So the purple means something specific structurally.
Exactly.
Yeah.
Gram-positive bacteria, the ones that hold on to that dark purple crystal violet stain, they have an incredibly thick outer cell wall made of this sugar protein mesh.
Wow. Okay.
Because that wall is so thick and dense, it acts like a sponge, soaking up the purple dye and just refusing to let it go.
Leaving us at the darks, which means the gram-negative bacteria, the pink ones, must have a completely different armor setup.
They really do. Gram-negative bacteria have a much thinner cell wall, but they sandwich it between two lipid membranes.
Wait, 2 layers?
Yeah, an inner and an outer lipid membrane. So when you wash them with the purple dye, that thin wall inside just can't hold on to it. The purple washes away, and they pick up the pink counterstain instead.
I can picture the armor difference perfectly. But let me push back a little here, playing devil's advocate for the listener.
Wait for it.
Why does it actually matter if a bacteria is a purple fortress or a pink sandwich? I mean, if a patient is sick, why does the color of the armor change how a doctor treats them?
Well, it changes everything about the battle strategy. That thick versus thin wall dictates completely different disease presentations and totally different vulnerabilities. Take the gram-negative bacteria, the pink ones with those 2 lipid layers. They contain something called endotoxins built right into their outer shell.
Endotoxins. That sounds bad.
It is. When your immune system attacks them and blows those bacteria apart, those endotoxins flood your bloodstream. It causes a massive, sometimes fatal, systemic inflammatory response called endotoxemia.
Oh, wow. So fighting them actually makes it worse at first.
Exactly. Your blood pressure tanks, your organs can fail. So knowing you are dealing with a gram-negative pathogen tells a doctor instantly that the bacteria dying might temporarily make the patient much, much sicker.
That's terrifying.
It is. And it dictates the exact type of antibiotic artillery you have to use.
That makes so much sense. You have to know how the fortress is built before you try to blow it up.
Right.
And alongside the armor, they're sorted by shape. You've got your balls, officially called cochi, and your rods, called bacilli. And then they're sorted by how they breathe.
Yeah, oxygen requirements dictate where in your body these pathogens want to set up camp. Aerobic bacteria need oxygen to survive, so they love the lungs.
Which makes sense.
Right? But anaerobic bacteria do not need oxygen. In fact, oxygen is toxic to them.
So they go underground, they hide out in deep tissue where the blood supply, and therefore the oxygen supply, is poor.
Like a deep puncture wound.
Obvious.
Which is why Clostridium species, which are strict anaerobes, cause things like gas gangrene. They thrive deep in necrotic tissue, happily fermenting and producing gas bubbles where oxygen absolutely cannot reach them.
That is wild.
And then you have the pragmatists, the facultative aerobes. They prefer making their cellular energy with oxygen because it's highly efficient. But if oxygen runs out, they just flip a metabolic switch and survive without it.
Okay, so we have our shapes, our armor stains, and our oxygen preferences. That seems like a very tidy sorting system.
It does, until you look closer.
Exactly, because as we comb through this material, there is a golden rule of bacterial exceptions that completely shatters that tidy system. It's like English grammar, I before E, except after C.
And in clinical deduction, spotting the rule breaker is often the key to saving a life.
The rule is this. Almost all of those spherical bacteria, the cochi, are gram-positive. They have the thick purple armor. Except for one notorious family.
Neisseria.
Yes, Neisseria. It's a gram-negative ball. It's the one exception, and it happens to be responsible for gonorrhea and severe meningitis.
So if a clinician looks at a spinal fluid sample and sees a pink ball, a gram-negative coccus alarm bells immediately go off.
Because it shouldn't be pink.
Right. That is Neisseria meningitis until proven otherwise, and the patient needs aggressive treatment immediately.
And the flip side of the rule is just as crucial. Almost all of the rod-shaped bacteria, the bacilli, are gram-negative, except for a very specific VIP list of gram-positive rule breakers.
The exceptions to the rods.
Yeah. Medical students apparently use the mnemonic ABCCCLLP to remember them.
Anthrax, botulinum, Clostridium, Corynebacterium, Listeria, Lactobacillus, and Propionibacterium.
That is a terrifying lineup. Propionibacterium causes acne, Corynebacterium causes diphtheria, Listeria contaminates our food supply, and Bacillus causes anthrax.
It's quite the rogues gallery.
Truly. So if you see a thick-walled purple rod under a microscope, you aren't dealing with a run-of-the-mill gut bug. You are dealing with one of these specific, often highly dangerous exceptions.
But if we are talking about rule breakers, the most fascinating ones abandon the rules of bacterial biology altogether.
Oh, the lazy ones.
Exactly. I'm talking about the obligate intracellular bacteria. This is where the strict line between a bacteria and a virus just completely breaks down.
Yes, bugs like chlamydia and rickettsia. These bacteria don't have that thick or thin cell wall we just spent all this time talking about.
They lost them.
And even crazier, they don't produce their own ATP. They don't generate their own cellular energy. They are biologically obliged to live inside human cells just to survive.
It is a combination of sheer evolutionary laziness and absolute brilliance.
How so?
Well, building a complex cell wall and running a metabolic engine to generate ATP that takes a massive amount of energy. Over millions of years, these bacteria figured out, why do the hard work?
Right.
Why build a fortress when you could just invade a human host cell, drop your bags, and steal their energy?
They are the ultimate biological moochers. They've outsourced their entire metabolism to us.
They really have. They stripped themselves down to the bare minimum genetic code required to survive and replicate. They rely entirely on our cellular machinery to do the heavy lifting.
Here's where it gets really interesting. Because those obligate intracellular bacteria provide the perfect bridge to the next major group of pathogens in our sources.
The viruses.
Yes. If those lazy bacteria are the biological moochers, viruses are the ultimate hijackers. We are transitioning away from living, breathing cells that consume nutrients into a territory of things so structurally simple, it's actually terrifying.
To appreciate a virus, you really have to understand the scale. They're incredibly tiny.
Like how tiny?
Most are less than half a micron wide. They are completely dwarfed by even the smallest bacteria.
The best analogy I could think of while digesting this material is that a virus is basically a malicious USB thumb drive.
I like that.
A thumb drive doesn't have a battery. It doesn't have a screen or a keyboard. It can't compute anything on its own. It's just a tiny core of data wrapped in a plastic casing.
Yeah.
And viruses are exactly that. A tiny core of genetic code, DNA or RNA, wrapped in a protein casing called a capsid. Sometimes they steal a lipid envelope to act as a disguise, but on their own, just floating on a doorknob, they are totally inert.
Right, they're not doing anything.
They don't do anything until they plug into one of our cellular computers.
That is spot on. They carry just enough code to force the host cell to read their instructions. And depending on what type of virus plugs into your cells, that malicious USB drive executes very different programs.
And our sources categorize 4 distinct ways these viral programs damage the human body. Number 1 is the lytic infection. This is the smash and grab robbery. The virus plugs into the cell, forces the cell to build thousands of new viral copies, and then simply blows the cell up to release the newly minted progeny.
Smallpox operates this way. Measles does too, but with a bizarre twist.
What does measles do?
Measles causes infected host cells in the lungs to actually fuse together into these massive, dysfunctional, multinucleated giant cells before it destroys them.
That sounds awful. Then you have the second method, non-functional.
Right.
The virus doesn't necessarily blow the cell to pieces immediately, but it ruins the cell's ability to do its actual everyday job. HIV is the classic example here.
Because it targets the immune system itself.
Exactly. It infects the command and control cells of our immune system, rendering them totally useless.
Leaving the door wide open for other infections, which we'll explore shortly. But the third mechanism of viral damage is perhaps the most tragic biologically.
This is the one I wanted to dig into because the mechanics are just so counterintuitive. It's called cell-mediated immunity.
It is a bit of a paradox.
In diseases like hepatitis B or infectious mononucleosis, which is caused by the Epstein-Barr virus, the virus isn't actually killing our cells at all. Our own immune system is doing the damage. Explain how that works.
Well, it highlights the brutal calculus of the human immune response. Imagine a hepatitis B virus entering a healthy liver cell. It sets up shop and starts quietly replicating.
Okay.
It might not actually destroy that liver cell's ability to function right away,
Yeah.
but our immune system, specifically the T cells patrolling the area, detect that the liver cell has been compromised.
Like it senses the hack.
Exactly. The cell is flying a tiny biochemical flag saying, "I've been hacked."
So the immune system calls in an airstrike on its own position?
Pretty much. It decides that containment is better than a cure. The T cells force the infected liver cell to commit suicide. Pathologists can actually see the visual evidence of this friendly fire under a microscope.
Really?
Yeah. If you look at a liver infected with acute hepatitis B, you'll see these structures called councilman bodies.
Oh, right. They look like these little pink ghost cells in the tissue samples.
That pink ghost is an apoptotic hepatocyte. Apoptosis is the scientific term for programmed, tightly controlled cell death.
Got it.
The immune system literally triggered a self-destruct sequence inside your own liver tissue.
So the horrible liver inflammation in hepatitis isn't the virus eating your liver. It's your immune system torching the village to prevent the virus from spreading. That is wild.
It really is.
And the fourth way viruses harm us is by causing cell overgrowth, sometimes called transforming infections.
In this scenario, the viral code plugs into the cell and hits the accelerator pedal on cell division. It forces the cell to multiply rapidly.
Just non-stop replication.
Yeah. This can lead to benign overgrowths, like the common warts caused by certain strains of the human papillomavirus, or HPV.
But constantly hitting the accelerator on cell division is incredibly dangerous. It's fertile ground for mutations.
Extremely dangerous.
And that's why these transforming viruses can cause full-blown cancer. Specific strains of HPV cause cervical cancer. The Epstein-Barr virus, the same one causing mono, can push B cells to divide so aggressively that it triggers lymphoma.
The virus essentially reprograms the cell into a tumor.
Unbelievable.
And this exactly. But the moment you experience intense physiological stress, your immune system's patrol weakens just a fraction. The virus senses the drop in security, wakes up, travels down the nerve, and creates a blister.
Your immune gardener took a coffee break, and the squatter went to work.
Perfect analogy.
And pathologists can actually identify which latent squatter is waking up by looking at the specific footprints they leave inside the nucleus of our cells. These footprints are called inclusions.
Right, the viral signatures.
Cytomegalovirus, or CMV, creates very distinct basophilic inclusions, meaning they stain a deep blue.
Whereas herpes simplex and varicella zoster create eosinophilic inclusions, which stain a bright pink. It all comes back to recognizing the microscopic clues left out by the hijacker.
So we've covered the independent fortresses of bacteria and the malicious USB drives we call viruses. Now we transition to the third major player in our sources. These are organisms that don't attack head-on.
No, they don't.
They wait in the shadows,
The fungi.
the great opportunists of the microscopic world. I love the analogy of fungi as pavement weeds. Like, if you have a concrete driveway, there are always tiny weed seeds sitting in the cracks.
Sure.
As long as the gardener is out there every single day spraying them and maintaining the yard, the driveway looks perfectly clean. But the absolute second that gardener goes on vacation or gets distracted fighting a viral infection on the other side of the yard, those weeds explode and take over the concrete.
That is the defining characteristic of clinical mycology. Fungal spores are everywhere. We breathe them in. They live on our skin.
Kind of gross to think about.
It is a little unsettling.
Hmm.
But they generally only cause deep systemic disease when the immune system, the gardener, is compromised or severely distracted.
Our sources break the fungal kingdom into layers of invasion. You have superstitial fungi, the dermatophytes that stay on the outer dead layers of skin, causing things like ringworm or athlete's foot.
The everyday annoyances.
Right. Then you have subcutaneous fungi. Then you hit the systemic ones, the deep three histoplasmosis, blastomycosis, and coccidiomycosis. The notes highlight that these deep fungi cause granulomas in the lungs.
Yes, they do.
Can we pause and explain what a granuloma actually is?
Well, it's an incredible defense mechanism, really. When you inhale the spores of those deep three fungi, your immune cells actually try to eat and destroy them.
Like a microscopic Pac-Man.
Exactly. But these fungi are tough. They resist being digested.
So what does the body do?
It shifts to a containment strategy. It sends a swarm of immune cells to physically surround the fungus, layering over each other like bricks, building a literal microscopic wall around the threat.
Oh, wow.
That spherical wall of immune cells trapping the fungus is a granuloma. It walls off the infection to protect the rest of the lung.
It's a biological prison cell.
It really is.
And just like bacteria had balls and rods, fungi have their own shape categories. We have yeasts, which are the round single-cell balls, and hyphae, which are long cylindrical branching tubes.
And this brings us to Candida albicans, the most prevalent yeast we encounter clinically. It targets moist, non-keratized squamous mucosa.
Meaning the wet tissues of the body that don't have a tough outer layer of dead skin cells.
Right.
So we're talking oral thrush, vaginal yeast infections, esophageal infections. Now, I noticed something interesting in the text. We aren't using the purple and pink gram stain to find these fungi.
No, we need a different approach.
The sources specifically mention using a PAS stain, which turns the fungi a vivid red. Why do we need a different paint?
Because fungal fortresses are built entirely differently than bacterial ones. Bacteria use that sugar protein mesh we talked about, but fungal cell walls are made of chitin.
Wait, like chitin, isn't that what bugs are made of?
Yes, it's the exact same rigid carbohydrate material that makes up the exoskeleton of a crab or a beetle.
That is bizarre.
It is. And the PAS stain, which stands for periodic acid Schiff, specifically targets and binds to complex carbohydrates. So when you apply PAS to a tissue sample, the chitin in the fungal wall absorbs it and lights up a bright, unmistakable magenta red against the background tissue.
That makes perfect sense. Now, the terminology around Candida is a bit confusing. The sources say it forms budding yeasts and pseudohyphae.
Ah, yes, the pseudo part.
Why the pseudo? Are they faking being those long branching tubes?
They are biologically faking it, yes.
Mm-hmm.
True hyphae, the ones you find in common molds, are long, continuous tubular structures divided by internal cross walls called septae.
Okay.
Candida doesn't actually grow true tubes. It just forms long chains of elongated yeast cells that stay attached to each other. Under a microscope, they look like long rods, but they lack those internal dividing walls.
Ah.
So clinically, they are pseudohyphae.
Pseudo or not, they are incredibly common. Leaning back into our gardener analogy, our sources establish what are essentially the 4 rules of fungus for a clinician diagnosing a
A
patient.
great checklist.
1. What shape is it? A yeast ball or a hypha rod? 2. Where is it? Superficial on the skin or deep in the organs? 3. If it's deep, is it triggering the immune system to build those granuloma prisons? And 4, the absolute most critical question.
Is the host immunocompromised?
Exactly. Which is the perfect bridge to our final section, because everything we've talked about today, the thick-walled bacteria, the latent cold sore viruses waiting in the nerves, the candida weeds on the driveway, They are all waiting for a specific crack in our armor.
They just need an opening.
And what absolutely blew my mind in these sources is the lock and key relationship between our immune system and these pathogens. Specific immune deficiencies lead to highly predictable, specific infections.
When you understand the mechanism of the defense, you know exactly what sneaks through when that defense falls.
Let's look at the first example, DeGeorge syndrome. This is a condition where a patient is born without a properly functioning thymus gland. The thymus is the boot camp where T cells are trained. Without it, you have a severe T cell deficiency. And the notes say these patients specifically present with severe candidiasis, massive overgrowths of the candida yeast.
Because T cells are the specific gardeners tasked with keeping candida in check on our mucous membranes.
The dedicated weed pullers.
Exactly. When the T cells aren't there to patrol the borders, the yeast encounters zero resistance and rapidly overtakes the tissue.
Then there's an even more specific failure, chronic granulomatous disease. Here, the patient has plenty of immune cells, but they are missing one specific enzyme called NADPH oxidase.
That's a crucial missing piece.
What does this enzyme actually do, and why does missing it lead to recurrent severe lung infections?
Think of your neutrophils, the first responder immune cells, as soldiers whose job is to eat bacteria. They engulf the bacteria, but to actually kill and digest it, they need to spray it with reactive oxygen species.
Like what?
Essentially, they manufacture microscopic bleach. NADPH oxidase is the chemical engine that generates that bleach.
Oh, wow.
If you are missing that enzyme, your neutrophils can eat the bacteria, but they can't kill it. The bacteria just survives inside the immune cell.
That sounds like a terrible strategy.
It is. So the body is forced to constantly build those granuloma prisons in the lungs to wall off infections it can't chemically digest.
That is fascinating. The soldiers swallow the enemy but forgot to bring their weapons.
Basically, yeah.
And the specificity continues with the complement system. The complement cascade is a series of proteins floating in our blood that act like biological hole punchers.
That's a great way to describe it.
They assemble on the surface of a bacteria and punch a physical hole through the cell wall to destroy it. The notes say a deficiency in the late-stage proteins, specifically C6 through C8, leads to recurrent Neisseria infections.
And that takes us all the way back to our bacterial rule breaker.
The gram-negative ball.
Right. Neisseria is that gram-negative coccus. It has a thin wall, but it has adapted to evade early immune detection. Its one true vulnerability is those late-stage complement hole punchers.
If you don't have them.
If a patient genetically lacks C6, C7, and C8, Neisseria essentially has a free pass to cause recurrent meningitis. If someone walks into a clinic with repeated Neisseria infections, a doctor can reverse engineer the problem and know instantly that those specific complement proteins are likely missing.
And then, finally, we have the scenario of broad immunosuppression, where the entire Gardner squad just quits.
Total collapse.
The total collapse of the system, most famously seen in advanced AIDS. The sources use a mnemonic for the flood of opportunistic infections that rush in. Stop catching IT.
It's a sobering list. Salmonella, toxoplasmosis, opportunistic pneumocystis, cytomegalovirus.
Wait, toxoplasmosis, that's a parasite, right? The one associated with cat litter.
It is. And many people actually contract toxoplasmosis at some point in their lives, and their healthy immune system quickly walls it off in tiny cysts, often in the brain or muscle tissue.
So you don't even know you have it.
Most of the time, you never even know. But when the T cells are wiped out by HIV, those cysts burst open. The parasite reactivates, crosses the blood-brain barrier unchecked, and causes severe neurological damage.
Which perfectly illustrates the terrifying reality behind the entire stop catching it mnemonic. All of these things, the latent CMV virus, the walled-off toxoplasmosis, the candida weeds, the tuberculosis bacteria, they are constantly present.
Just waiting.
They are already inside you or lingering on the surfaces around you. They are only kept at bay by a fully functioning, highly complex immune shield. The moment that shield drops, the entire microscopic war zone spills over the walls.
It's a delicate, ongoing balance of power that keeps us alive every single day.
So what does this all mean for you? Why do we just spend this time exploring thick bacterial walls, viral USB drives, fungal chitin, and missing enzymes?
Good question.
Because understanding these microscopic rules fundamentally transforms the way you look at illness. It stops being just bad luck. It stops being a mystical, random ailment. It becomes a highly choreographed biological interaction. You can visualize the mechanics. You understand that a gram stain isn't just a color, it's a map of a pathogen's armor. You know that a cold sore is a squatter waking up when your guards are down.
a weed on the driveway.
You realize that a fungal infection is just a weed taking advantage of an absent gardener. It removes the mystery from medicine and replaces it with logic.
It gives you the blueprint to the battlefield. And, I want to leave you with one final, slightly provocative thought to mull over.
Let's hear it.
Early on, we talked about those obligate intracellular bacteria like chlamydia. We discussed how they lost their complex cell walls, gave up their ability to make their own energy, and evolved over millions of years to live entirely inside ourselves, relying on our machinery.
The ultimate moochers.
Exactly like a virus does.
Yeah.
It makes you wonder, if a complex living bacteria can devolve and strip itself down to become so incredibly virus-like, could viruses eventually evolve to become more like bacteria?
Oh, wow.
When microscopic entities are constantly trading genetic parts just to survive inside us, where's the actual definitive line between what we consider a living organism and a rogue piece of genetic code?
A diagnostic muddy water indeed. Thanks for taking the deep dive with us.